Greetings, students. I am going to talk about how you control what goes in and out of a cell when it's large and polar, or just really large, because it ends up being a slightly different scenario. And the way that that happens is endocytosis and exocytosis. So end means in, X means out. And we'll talk a little bit too about why cells are small, but this should be a shorter presentation. All right, so when we're talking about endocytosis, there's kind of three ways of talking about it. And the first two, but they're basically the same. I'm all right with the, I think the distinction's kind of academic, so it doesn't really matter. But you have phagocytosis, and this really is cell eating. And what happens is the cell, so here's the cell is going to sense that there is something on the outside. So you would have a receptor-mediated protein that would sense that there's something out there. And then what's going to happen is the cell is going to start to, so let's just say, find a little thing, this little red blob, and the cell wants to have that inside it for whatever reason. When that occurs, and the cell is going to start let's get rid of the red. That'll be black. This is not the easiest. The cell is going to start to change shape. And as the cell changes shape, it's going to, so this is kind of a path here in time. First the cell looks like that, then it looks like that, and then finally the cell is going to look like that. And this object is going to stay inside it at each time. So the cell is going to change shape, and as it changes shape, it's going to surround the object. So that is called phagocytosis or cell eating. It can happen when there is some type of particle, could be a food particle. That's really for single-celled organisms. What we end up seeing this the most for in multi-celled organisms is white blood cells, the immune system. The cell can also do this if it's really important that it brings in a bunch of fluid. So this is cell drinking. I don't really care if you pay attention to that. But the other one that I want to point out is this idea of receptor-mediated endocytosis. So this is actually a little bit of what I was describing, because cells have to know that something's going on. But in this example, you have a very specific receptor on the outside of the cell membrane. And when that receptor binds with another specific molecule, this little purple triangle, then it's going to cause this to happen. So it's going to cause this indentation, also called an invagination, to happen, so that you get this to go in. Now you're like, Mr. Levante, why do I care about this? I'm so glad you asked. The reason you might be interested in what a ligand is and a receptor is one of these receptors that we have on our body is the ACE2 receptor. The ACE2 receptor is the receptor protein that the SARS-CoV-2 virus joins to. And so the way that SARS-CoV-2 gets into our cells is the spike protein, so this would be a spike protein, binds to the ACE2 receptor, which is there actually to deal with hormones and water regulation normally. But the spike, SARS-CoV-2 takes advantage of it by having a ligand, or this little thing that looks like it, and it causes, this is how SARS-CoV-2 gets into our body. So this is via endocytosis. This is just a description of pinocytosis. Yeah, actually, so you can see this happening in like the small blood vessels when you're trying to move a lot of water in and out quickly. What is created is a vesicle. So we're going to talk about this a little bit in another video. But what that means is that you have a phospholipid bilayer here, right? So that's what that is right there, because this is a phospholipid bilayer, and so that's a phospholipid bilayer. All right. One of the kind of a super interesting things about this idea of having endocytosis happen is that one mystery that we've had in biology is, how did we move from a prokaryotic cell to a eukaryotic cell? All right. So I'm going to define a couple of these terms. Prokaryotic is, it actually literally stands or means before the kernel, or before the nucleus. And then eukaryotic, U is with, that is with the nucleus. And so how do we get a nucleus in the cells? And this woman, Lynn Margolis, a super famous scientist, came up with this theory that, in fact, the way we got a nucleus was that via endocytosis, that there was another cell, another prokaryotic cell, that had a bunch of useful DNA, and there was a symbiotic relationship between that cell, so that's the DNA cell, and then we'll call this one, I don't know, Lynn, and another cell that was using that DNA. So the DNA was being used by this cell, and this cell was probably giving that cell food. And then eventually what happened is endocytosis happened, and this DNA cell, so I'll make it black again, this ended up being the DNA cell, and then this cell ended up being the red cell, or the parent cell. There's some evidence of this. Obviously, this happened a long, long time ago. I'm trying to think, I think 3 billion years ago. Hold me to that. But somewhere around there is when we started seeing the first eukaryotic cells. But another interesting thing is there is a double lipid bilayer here. And remember, if this is going to have, so let's see if I can zoom in here. If this cell is going to have a lipid bilayer, so that's 2 layers right there, and this cell is going to have a lipid bilayer, when it goes in, right, then what you should expect is that you would have 2 layers here and 2 layers here, right? A double lipid bilayer. And that is, in fact, what you have in the nucleus. Additionally, it is thought that the mitochondria, so I'll draw universal sign for mitochondria there, the mitochondria, and in plants, chloroplasts also went into cells, went into eukaryotic cells through endocytosis. So that got absorbed and that got absorbed. Both of these organelles have their own DNA, and both of them have a double lipid bilayer. So that's true of both of these organelles. So that both of these were prokaryotic to begin with, but then got incorporated and now are organelles in a eukaryotic cell. So I thought that would be kind of an interesting way to end this concept of business and exocytosis. Here is this is an example of how this works. So this is showing you it step by step with cyanobacteria. This would be the original chloroplast, what eventually became chloroplast, and then this protobacterium, which became mitochondria. And this is all Lynn Margulis's theory. All right. And then this is kind of a specific example of really what's going on with these specific organisms. So it's showing you how this can happen beyond chloroplast and mitochondria. These are these methanogen cells that use methane. So I'm not going to go into that. Okay, so this is evidence for it. We've talked mostly about this, but new mitochondria and chloroplasts are made by binary fission. So this is how prokaryotes divide. So they're using the same mechanism that prokaryotes use in order to reproduce. So this is a specific type of cell membrane. You have a cardolipin in there and a transporin. Okay. No eukaryotes don't show any of this, but all prokaryotes do. So I'll highlight that, and then I'll put a little dot here, and I'll write prokaryote. So that's a prokaryotic trait. I already mentioned that they have their own DNA, and that DNA mimics or looks more like prokaryotic DNA. And they also have their own ribosomes. So ribosomes are a combination of proteins and RNA that are used to make proteins. Generally, there's no other organelles in our body that have their own ribosomes except for mitochondria and then chloroplast in plants. And these ribosomes are more closely related to prokaryotes than they are to eukaryotes. So all of this is evidence for the endosymbiont theory. All right, we're going to move on to one more idea, and this is surface area to volume ratio. So this starts just with a little bit of math, which we'll skip. You can think of cubes and rectangular prisms and how you're calculating volume, right? Length times width times height. But what really matters here is this idea that if you change, so what I have down here, these are just base units. It doesn't even really matter what they are, but you could call them, let's say, millimeters or something like that. So as you move in size, in diameter, so this is R. So on the bottom you have R, okay? And then, so this is a radius. And then over here you have basically two different values. The blue line is surface area, and the red line is volume, okay? And what I want you to see is how these two values change in relationship to one another. So as you get larger, surface area increases in a linear surface area is linear, and volume is exponential. So the volume increases much, much more quickly than surface area increases. Now, since a cell, so I'm going to draw this here because I'm not going to have a lot of room. Since a cell relies on diffusion primarily to move material in, so let's just take the obvious example of oxygen and out, carbon dioxide, right? So carbon dioxide is going to be going out, O2 is going to be going in. The mechanism for this is diffusion. Since the cell relies on diffusion in order to have that happen, it's essential that cells remain small. And let me try to draw this. So let's just say that this is a normal size cell and this is working fine for this cell, okay? And let's pretend here, I'm going to enlarge this. Let's pretend that whatever this distance is, let's call it x, right? And let's say it takes, whatever, 30 seconds. So for a given size, all the little spots in here are going to need to get oxygen. And for a given size, this carbon dioxide is going to have to get out here. So hopefully you can see that, so whatever this is, let's say it takes 30 seconds to move this distance if you're an oxygen molecule and you're trying to get in the cell. So this is the radius. It takes 30 seconds to get there. So what I hope you can see is if you had a cell that was larger, it would still take 30 seconds to move the same distance, right? So this too is x. That's going to equal x. Well, what that's going to mean is you're only going to get oxygen to diffuse this distance. And carbon dioxide is going to get trapped in here. So there is going to be a limit to cell size because cells require this passive movement of oxygen and carbon dioxide for diffusion. At a certain point, the inside of the cell is not going to get any oxygen. It's not going to be able to get rid of the carbon dioxide. And it's going to die. So the reason cells are small is because they rely on this concept of diffusion. And then finally, I found it helpful just to make a distinction between cell walls and cell membranes. It's important to know that a cell wall is structural and permeable. So it does not try to keep the good stuff in and the bad stuff out. The only thing it does is keep structure and make it stiff. And as I bet you can guess, it is made of cellulose. E-L-L. I can't write it there because I don't have enough space. So I'll review all of this in class, but I wanted to give you just a little sense of that. All right. Take care.